I am sure you have heard about nucleic acids. Even my grandmother has, I believe, because this is on the news all the time. You've heard about DNA, you've heard about RNA, not as famous as DNA, but it is quite common. Even in basic biology, you have talked about these molecules, these class of molecules. But here in this tutorial, I want to go into some sort of detail enough for you to go through medical school or classes like molecular cell biology. And the first thing I want to mention is that these are biological molecules or organic molecules, however you would like to designate them. And they're essential for life. If you didn't have them around, life would not be possible. And this is the same thing for lipids, for proteins, for carbohydrates, so on and so forth. The second thing I would like to mention and to keep in mind, second thing on this first slide so you can remember later on when we talk about this, is that these are linear molecules which consist of nucleotides as the monomers. Okay, so the units here are nucleotides, and we're going to discuss that later, the structure of this monomer. Third thing I want to mention, and this is what distinguishes this class of molecules from others like proteins, like lipids that have other functions, that these are the molecules that are able to store and express genetic information. So your eye color, the color of your hair, diseases that are transmitted from one parent to the offspring, this is thanks to these molecules. When you talk about nucleic acids, you have to think that there are two major classes or subclasses of molecules, let's call it. And the first one is DNA, abbreviation for deoxyribonucleic acid. Sounds like a long word, but it's not. It's really not. And when I decompose it and later on and talk about it, you're going to see why we have this word. It's a very simple reason. But it is important to mention that DNA, this molecule, is responsible for storing the genetic material, genetic information. So everything about your height, the color of your eyes, anything you inherited from your parents or your family, this is stored in the DNA. Never forget that. That's why it's quite famous molecule, let's call it. But there is another molecule that, even though less famous than DNA, still quite important in your cells. It's called ribonucleic acid, or RNA, and this is a very important molecule because it is involved in various cellular activities that we're going to discuss and even go through some classes of and different types of RNA that exist in your cell, many types actually. And it also catalyzes some reactions, and we're going to see some examples of that later on. So when I talked about the nucleic acids, I mentioned that the building blocks of these molecules, or the monomers, are nucleotides. And if you look individually at a nucleotide, you are going to find three distinct structures. And these structures are a base, you're going to find a base, a pentose in the middle, and phosphates, which can vary in a nucleotide, keep in mind, can vary between 1 and 3. Okay? So if you want to impress your molecular cell biology professors, you should definitely know how to draw a basic structure of a nucleotide. And you can start with the pentose in the middle. And you're going to have a base on one side like this, and 3 phosphates. I'm going to draw the maximum. This is the maximum capacity, let's say, of phosphates on this molecule. And here I have my pentose. And keep in mind that a pentose is a sugar. So I'm going to write here, pentose is a sugar, and we're going to deal with that later on when I post a tutorial on sugars. Keep that in mind. The first area, or the first structure I want to discuss here in this molecule, the nucleotide, is the pentose, this 5-carbon sugar that we're going to discuss in later tutorials when we talk about carbohydrates. But right now I want to tell you that there are 2 major types of pentoses in nucleic acids. And the first one is ribose, and the other one is deoxyribose. And I'm going to draw the structures for you so you can understand the major difference between them. And as you can see, they're very, very similar, basically the same, but one major difference. If you look at ribose, you have 2 hydroxyl groups. And if you look at deoxyribose, there is only 1 hydroxyl group. So 1 less oxygen, as you can see. And ribose is found in RNA because, as the name indicates, ribonucleic acid. So ribose for ribonucleic acid. And deoxyribose for DNA. So this pentose is found in DNA for deoxyribonucleic acid. And deoxy means 1 less oxygen. Deoxy, 1 less oxygen, as you can see here. So keep that in mind. Another important thing to discuss about these molecules is that they have 5 carbons, and these carbons are very important to be numbered, or so you can understand the orientation of these molecules. So I'm going to number them 1 to 5, 1, 2, 3, 4, and the 5th one here. So now I want to talk about this structure right here that you can see, the base in your nucleotide. We just discussed the pentose. Now it is time for a more complex type of molecule, let's say, that we see in the nucleotide, which is a base. And the bases are heterocyclic, which means that, well, the name says it all. Hetero and cyclic means cyclic ring, and hetero, there are at least two or more different types of atoms in this molecule. But in this case, in the nucleotide, the bases in the nucleotide, you are going to find 2 types of atoms, nitrogen and carbon. So they are comprised of nitrogen and carbon rings, like I'm writing down now. A second thing I would like to mention, remember when I started numbering down the carbons in the pentose, it is important. Here's another reason why it is important to number those carbons, because the position to which the base will bind in the pentose is called position 1. So carbon number 1, and it's right here. As you can see, this is where my base is going to bind in the nucleotide. Carbon number 1, you have it here. And the third thing I would like to mention about bases is that you can find 2 types of bases. One is called purines, and purines have 2 types of molecules. One is adenine and guanine. And these molecules have something in particular. They actually are comprised of 2 rings, 2 different rings. We're going to look at the molecules later, maybe on the next slide. You're going to see them. They look kind of like this, 2 different rings. Okay, and the second type of molecule is pyrimidine or pyrimidines, and you find 3 types of pyrimidines. You find the cytosine, the thymine, which you only find in DNA. You don't find thymine in RNA. Instead of thymine, you're going to find something else substituting this. Let's say in RNA, you find uracil, another type of pyrimidine. So in RNA, uracil. Thymine only found in DNA. And they kind of look like this, like an hexagon. As I promised, I was going to show you the actual structures of the bases in the nucleotides. So this, we're still talking about this structure here in the nucleotide. And as I mentioned before, we have some structures called purines as bases and others pyrimidines. And the purines, as you see here, they have 2 rings. One of them is in shape of hexagon, so it's hexagonal, and you can see here, like I'm showing, and the other one looks like a pentagon, something like that. I'm not a very good drawer, and this is the one that I'm talking about. And these two, we have two types of purines, like I mentioned before. And this one here is called an adenine. And from now on, we're going to mention it or abbreviate to an A. When we talk about nucleic acids, the A stands for adenine for this base that you see here in the nucleotide. Okay, so keep that in mind. And this one right here is going to be my guanine. That's the other type of purines that we talked about. And the abbreviation for it in nucleic acids is a G. Okay, so every time we have a G, I'm talking about a guanine in this structure here of the nucleotide. Okay, and for the pyrimidines, we have this structure, only one ring of the shape of hexagon with 6 corners, and as you can see, this is the shape I'm talking about. Okay, and as I mentioned before, we have 3 types of pyrimidines, and this one that I have here is a thymine. So thymine, and every time I talk about it in nucleic acids is going to be a T in this area here in the base. This one here is my cytosine, and abbreviation for C. These two will be found in DNA. Never forget DNA, DNA. Okay? Meanwhile, you're going to see, instead of thymine, you're going to see this one here, uracil, in RNA. This is for RNA only. You will not see uracil in DNA, nor you'll see thymine in RNA. Keep that in mind. So this one, DNA. And the abbreviation for uracil is a U. So every time you see a U, you know it's going to be a uracil in your base here. So now it's time to talk about my phosphate groups that you can see here. It can either be 1, 2, or 3 in a nucleotide. You can find these variations in a nucleotide. I'm going to write down phosphate groups, and I'm going to draw a molecule so you can have an idea of what I'm talking about. And a phosphate group is basically a phosphorus atom surrounded by 3 hydroxyl groups right here, 3 hydroxyl groups, and 1 oxygen on the top that I just threw there. And another important thing to mention about the phosphate groups is that they can bind to either the 5' position or the 3rd' position. Or what I mean by this is another beautiful way to say 5th position or 3rd position or 5th carbon or 3rd carbon on the pentose that I'm going to draw in the center. And here is my pentose, and the carbons I'm going to number. So you have, remember, 1, 2, 3, 4, and my 5th carbon right here, this little tip here. Okay, so, and what is going to happen? I'm going to be able to attach a phosphate group here, and sometimes you're going to see that they're able to attach to the 3rd position as well. So keep that in mind, and you're going to see that later. I'm going to explain how this happens. Meanwhile, I want to explain a word that you might hear some other time, nucleoside. You're used to nucleotide. I talk about nucleotide all the time because I usually include the whole thing, the whole package, the base, the pentose, and the phosphate group. But a nucleoside lacks phosphates, so lacks the phosphate groups that I just mentioned. So whenever I talk about a nucleoside, I'm definitely not including a phosphate group, just the base and the pentose. Okay, so keep that in mind. I want to go quickly over the phosphodiester bond that you found, or the bonds you find in nucleic acids. This is how you go from a nucleotide binding to another nucleotide, binding to another one, and forming a polymer. The polymers we know, the DNA, RNA, and nucleic acids, like we have been talking. And I have a very simple illustration here which will probably help you understand what I'm talking about, the phosphodiester bond. And the first thing you need to understand is that we have two nucleotides. They are going to bind to each other, and this is how it happens. Start counting 1, 2, 3, 4, 5. The carbons, okay? Another counting 1, 2, 3, 4, 5. 5 carbons on each nucleotide, on the pentose, I mean, okay? And another thing that you need to know is that if you look here, the hydroxyl group bound to this third carbon, okay, in the third position, you can call it 3 prime, okay, is going to react with the phosphate bound to the carbon on the 5th position, or the 5 prime position. Okay, this is it. This is what is going to happen in the 3 prime to 5 prime phosphodiester bond. That's why you name it 3 prime to 5 prime. Okay. Oh, sorry. Let me correct. Okay, there you go. So this is how you get this. And I have a better illustration here. Hopefully a little bit better, not too much, but enough for you to understand. And here you have one nucleotide, another nucleotide, and you have your pentose, base, and phosphate groups, okay? Same thing with the carbons, but I'm going to write here, this is my 3rd carbon here, okay? And this is my 5th carbon, okay, on the 2nd nucleotide. And what is going to happen, this phosphate group here, with, you can see the oxygen here, prepared to react with this hydroxyl group here on the 3rd position and 3 prime position, and here on the 5 prime position, the phosphate, okay? This reaction will produce H2O molecule. Hopefully you know what it is. It's a water molecule. And here you have the result. 2 nucleotides bound to each other. And my phosphodiester bond is right here. You can see it here. This is my phosphodiester bond. A carbon, 1 oxygen, 1 phosphorus, 1 oxygen, another carbon. This is my phosphodiester bond, 3 prime to 5 prime, because of, I've just explained the reason, due to the carbons that are going to be involved in the reaction. Okay, and not involved, but let's say that where these molecules that are important components of the reactions are attached to or bound to. That's a better way to say. So another thing I would like to explain here is why I have a 5 prime end and a 3 prime end. Think about when this molecule is forming. Imagine that we have 15 nucleotides bound to each other in this fashion. What is going to happen, if you look at the end of the molecule, there is 1 carbon here that is not interacting with any other molecule, okay, except the phosphate group here, but usually no interaction. And this is my 5th carbon, so this is a 5 prime position, 5 prime end. This is why I have this here, okay, this nomenclature, let's say. And I call it 3 prime end on this side of the molecule because this end of the molecule, I have a carbon in the 3rd position, 3 prime position, that is not interacting with any other nucleotide. Except you see here that it's a hydroxyl group, but there is no other interaction with any other nucleotide. That's why I call it a 3 prime end. Thought nucleotides were just involved in the composition of DNA and RNA. Like if you thought they were only monomers for these two big molecules, you are wrong. There are actually other forms of nucleotides in your cells, and I have two examples here that illustrate what I'm talking about. The first one is ATP, and I'm writing down the structure, the basic structure of a nucleotide on ATP, on this ATP molecule that I have here, so you can understand what I'm talking about. So you have adenine as a base, the pentose right there, the 3 phosphates right here, 3 phosphates in ATP, because it's adenosine triphosphate, okay, due to the base and the 3 phosphates that you see there. Another molecule that is an example of other functions for nucleotides is a CAMP. And CAMP stands for cyclic, so cyclic adenosine monophosphate. In this case, we're not talking about triphosphate because I'm going to explain a little bit later why the mono and triphosphate, but this case, monophosphate, because you only see one phosphate group. And you see the pentose here, the sugar, the base of the nucleotide. In this case, we're talking about an adenine as well. Adenine for a base. And at the end here, you see a phosphate group, which is bound to the pentose in a fashion that looks like a ring, and that's why you have cyclic in the name. And energy storage is definitely the function for ATP. I'm sure you have heard about it. It's a very, very famous molecule. Everyone talks about ATP when they need energy. I need energy all the time. And usually I go for coffee, but it doesn't work that way. Anyway, but CAMP actually has a regulatory function. Another example is GTP. We're going to see it later on. Regulatory functions in your cell. So another function for nucleotides. The last or another thing that you can see nucleotides doing is that they are components of coenzymes. And two examples are NAD and FAD molecules. You're going to see them in later topics. I believe it is important to understand how things are named, especially nucleotides. How do you name a nucleotide? And you name it according to the base, okay? So I have a quick table here that I'm just writing down. So for base, if you have as a base an adenine, your nucleoside, or in case you have phosphate groups, your nucleotide is going to be named adenosine. If you have a guanine, it's going to be named guanosine. A thymine is going to be named thymidine. Uracil, uridine, and cytosine, cytidine. Okay? And I can give you an example that I just mentioned previously, but just a reminder, ATP, adenosine triphosphate, because in the molecule the base is an adenine. Okay, that's why you name the nucleotide adenosine. Okay, when you name a nucleotide after a base, like I just explained in the previous table, you also have to do it according to the number of phosphate groups. And if you attribute letters, say this is the alpha, middle phosphate is going to be a beta, and the one furthest away from the pentose is called a gamma phosphate group, you can see that a nucleoside with one phosphate group is called monophosphate. You add monophosphate to the actual name of the molecule. And a line of molecules I'm going to use to give you an example is AMP, for example. And this is adenosine monophosphate, meaning that the base is an adenine, but in the molecule there is only the alpha or only one phosphate group. When you have a nucleoside with two phosphates, you have a diphosphate. This is when you have the alpha and the beta. And an example taking the same class of molecules, the ADP, adenosine diphosphate, meaning that you still have the adenine for a base, but you have two phosphate groups, the alpha and the beta. When you have a nucleoside and 3 phosphates, this is the maximum of phosphate groups you can have in a nucleotide, triphosphate. And an example, the very famous molecule that I just talked about, ATP, responsible for energy storage in your body, in our body, I'm human too, is adenosine triphosphate, also with the adenine for the base.